Hey class, Adam Ward here. I want to talk with you today about BOD, or biochemical oxygen demand. And so this is all about how the organic matter in the water is going to be degraded and in doing so consume dissolved oxygen out of the water column. So if we're thinking about our conventional wastewater treatment plant, the portion that we are going to be interested in here is happening over here in the secondary treatment. And in fact, it's in this, what we would call the aeration basin. And so you can see where I'm drawing on the screen with red here. What I'm trying to convey to you is that we've got this basin where we are going to use pumps to bubble oxygen, typically just air from the atmosphere, in through the water column. That O2 will get used as part of microbial growth, so there'll be respiration. And the consequence is that CO2 will ultimately bubble out of this portion of the system. And what we want to focus on in this video is exactly what happens that is consuming that oxygen and how can we plan for that in our system. Okay, so BOD, biochemical oxygen demand, writ large, is the amount of oxygen that will be used by microorganisms as they oxidize organic matter. In other words, these microorganisms are going to break down organic matter to derive energy from it, and as they do, they're going to respire, just like you and I are doing right now. So we will take oxygen in our case, into our lungs. For a microbe, they'll be using oxygen from the water column as part of the reactive process. And CO2, what we are breathing out, or what will be bubbling out of our aeration basin, is a consequence of that transformation. And so as we think about our BOD, the plot on top is meant to describe what's called BOD exerted. So how much of that oxygen demand have we seen so far? And on the bottom is more what we're accustomed to seeing, which is sort of the inverse, or the mirror image across the horizontal axis, how much BOD is remaining. Now, as we study BOD, one of the really important things that we'll measure is BOD5. And what this simply means is, if we think about a first-order reactive process taking place, BOD5 is telling us how much oxygen gets used within the first 5 days. Now, it's not that the first 5 days are critical in any way, shape, or form. It's that 5 days gives us enough transformation that we can project forward. So we could use that knowledge to find out how much BOD was there, what is the characteristic transformation rate, that first order coefficient K, for example. And we can use that measure to make some forecasts of what we call BOD ultimate, or BOD with a subscript U after it. And this is meant to be this exact same first order process, but let run entirely to completion. So as time goes to infinity, how much BOD will be exerted, or how much BOD will be realized. At the ultimate BOD, there would be no organic matter left in the water column to be decomposed. All of it would be decomposed, and in turn, all of the oxygen that would be needed will have been used by that time. This is just a quick introduction. In our class, we're using the textbook by Mines, and section 3.3 of that textbook gives a nice primer on BOD. So if what I'm talking about here isn't making a lot of sense for you, or if you want some additional resources, check out 3.3 in our textbook. Finally, just a note that organic matter being degraded by microbes is perhaps the largest source of oxygen demand that we'll deal with, but it's not necessarily the only one. So in addition, we often describe chemical oxygen demand, or COD, and you could think of this as a totally inorganic process. So if there aren't microbes involved, it's simply a chemical reaction that consumes oxygen. You'd put that in your COD category. And finally, we talk about a theoretical oxygen demand, and this is the idea of how much oxygen would get used if all of the input, both the degradable organic matter and the chemicals, would be decomposed and ultimately leave our system, ultimately take as much oxygen as they need out of the system. So from a chemical reaction perspective, BOD is sort of a two-step process. And to be really clear here, we're going to talk about two kinds of BOD, one associated with carbon and one associated with nitrogen. So we call those Cbod and Nbod. And just to make life confusing, that is not the same as COD. So COD is chemical, Cbod is carbonaceous BOD, and Nbod is nitrogenous BOD. So I've drawn just a simplified reaction here in the black text, but what I'm trying to represent to you is the idea that organic matter in our water column is reacting with dissolved oxygen, being respired by the microbial community present to generate new biomass and the respiration byproducts CO2 and water. And it's this use of dissolved oxygen that we really care about. So one of the consequences that we environmental engineers realized early on about releasing our waste into nearby lakes or streams is that we were decimating the ecosystem. And one of the big culprits was the organic content. So as that organic matter degrades, oxygen gets stripped out of the water column, and we're essentially preventing the aquatic life from existing there. This is entirely parallel to what's happening in the Gulf of Mexico as a result of nutrient pollution that fuels large algal blooms. That's a source of organic matter. And then as those algae die and sink to the bottom, they decompose, and that decomposition is stripping oxygen out of the column, and we call that the dead zone in the Gulf of Mexico. Okay, so we've got our carbonaceous BOD, and when we decompose that organic matter, we won't get 100% of it turned into CO2 and water. As an example, there will be other functional groups, like amino groups, which are NH2s, that will still exist. And with nitrogen in particular, we'll go through a series of steps that will lead us to the production of ammonia in the water column, so NH4 with the +1 charge. And you can see that's where the story of Nbod now begins. So just like we see for carbonaceous BOD, there will be a microbially mediated process that is going to use dissolved oxygen out of the water column as part of this transformation. And so you can see I'm highlighting there in red those two O2 molecules. So as our ammonium is nitrified, we'll also use oxygen out of that water column. So if we think about how these are going to look through time, the Nbod exertion, so when that reaction is actually using oxygen out of the water column, tends to be a little bit delayed compared to carbonaceous BOD. Notice our BOD5 that we looked at really early on is primarily measuring the carbonaceous component. And so if there's going to be significant Nbod, it wouldn't be captured in a 5-day test. In that case, we often use a 30-day test where we can start to capture some of that Nbod as well. So 30 days being the incubation time in your laboratory, for example. So the lag that we see is essentially because this is a sequential process. So we need to have the Cbod get exerted and generate those amino groups, and then we need the microbes that are involved in the nitrification process to be able to reproduce in sufficient numbers so we actually realize that Nbod. So the reason for that delay is largely because it's a sequential process. And if we go out far enough, we can see the ultimate Cbod, the ultimate Nbod, and the ultimate BOD overall that would be exerted. And that would be where those lines all start to level off on the right-hand side of this plot. So the last thing I wanted to do on BOD is a quick example, just to help you see how these calculations might look. So in this problem, we're given wastewater with 100 milligrams per liter of ethylene glycol, chemical formula given, and 50 milligrams per liter of ammonia as N. And we're asked to determine the theoretical Cbod and Nbod, and I'll also do the ultimate, the total BOD for you as well on the next slide. So I'm going to start with the Nbod component, and I just want to make sure that we help translate this for you. So we're given what we're given here is 50 milligrams per liter NH3 as N. What that means is there are 50 milligrams of nitrogen per liter, and all of that nitrogen is in the molecular form NH3. And I think the calculation will help clarify this a bit. So written in gray is the key equation that we're looking at here for nitrification. So starting with our 50 milligrams of ammonia as N per liter, we're going to go ahead and turn that into grams of N. And then we know the molecular mass of nitrogen is 14, or the atomic weight of nitrogen is 14. So 1 mole weighs about 14 grams. We can use our knowledge that this was given to us as N to convert it into molecules of NH3. Our balanced equation tells us that for every mole of ammonia that we're going to process, it's going to take 2 moles of O2. 1 mole of O2 is 32 grams of oxygen, and that's an easy spot to make your mistake. If you were thinking, hey Adam, why isn't that 16? 1 molecule of oxygen, or 1 atom of oxygen has that atomic mass of 16. O2 contains 2 of those. We need to double that, 32 grams of oxygen in 1 mole of O2 molecules. And finally, we can convert this back into milligrams, so milligrams per liter being a typical concentration that we'll use. Always good to go through and cross out your units to make sure that you see you're left with milligrams of oxygen on the top, liters on the bottom, and I got a total of 229 milligrams per liter of Nbod, or nitrogenous biochemical oxygen demand. The story's pretty similar for Cbod. So here's a really generic equation written for our ethylene glycol. Always good to check that this equation is actually balanced, and so you can see I've added those components out front. And so at this point, rather than giving you the solution, what I will do is tell you, using a similar workflow that we saw on the previous slide and stoichiometry, which I think you all are pretty good at by this point in your careers, we should be able to get from 100 milligrams per liter ethylene glycol to about 129 milligrams per liter of carbonaceous BOD. With those 2 calculations done, we can also look at our theoretical BOD ultimate. In other words, what's the most oxygen that could possibly be used if both Cbod and Nbod are 100% realized? That's as easy as adding the 2 solutions we had together, 229 milligrams per liter from the previous slide, 129 from this slide, and I got a total of 358 milligrams per liter of theoretical BOD. All right, folks, so hopefully this recording helps you understand a little bit about what's meant by BOD, why it exists, and how you could use that knowledge to understand how much oxygen you're going to need to supply to your aeration basin if you're going to let the microbes respire and decompose or remove that organic matter from the water column. All right, thanks everybody. Hopefully this helps, and I will see you back in lecture.